Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the genetic code. Okay, so before going to this explanation, I want to say that I have already explained the translation process and in my previous video, so you just check out that. Okay, and another thing is that if you are the newcomers, so please and if this channel is helpful to you, so you just share and subscribe this channel and give a comment obviously in the comment box if this is usable or not. Okay, so first is thing that the genetic code. What is the genetic code? Genetic code is the triplet code and this is called the codon. Okay, genetic code is the triplet code and this is called the codon. So without codon or without presence of codon, the translation process never be started. And there are two types of codon. Another is the initiation codon and another that is the stop codon. Initiation codon is what? The presence of initiation codon, the translation process that begins, and the presence of stop codons, the translation process that becomes stopped. Okay. So main initiation codon is AUG and this specifies the methionine. This is the amino acid and this specifies the methionine. Another important stop codons that are UAA, UAG and UGA, and these generally presence of these stop codons, that translation becomes stopped. Okay, so here another thing is that beside this AUG as the initiation codon, there are another initiation codon also present, that is the GUG and the UUG. Okay, these also acts as initiation codon. And generally one triplet codon that specifies one single amino acid. And if the translation process become continuing, so the codon that comes continuously. Okay, so that's why the codon has no comma. These are the commas. Because if the translation process continually, they continuously passing for binding the amino acids, and that is the code is degenerate. What is the degeneracy of the codon? That is one amino acid that can be specified by more than one triplet codon. That means more than one triplet codon that can bind one amino acid. Clear what I am saying? That more than one triplet codon, or you can say that more than one codon that can bind one single amino acid. Single means only particular amino acids. Like that UUU and UUC, these are the synonyms for which phenylalanine. Okay, that means if UUU, that codon is present, so the phenylalanine amino acid that can be bind. Another way, UUC, that codon is present, that means again phenylalanine can be bind. Okay, so this is called the degeneracy of the codon. Okay, so in generally 18 of the 22 amino acids are said to be synonymous, and this property of the codon, that means the degeneracy of the codon, this property of the codon that binding the one amino acid, or the you can say that more than one codon that can easily bind with one amino acid, this is the feature of the codon, and this feature is due to presence of this feature within the codon, so these codons are called the synonymous. Okay, these codons are called the synonymous. Next thing is that the code is non-overlapping. One code is not overlapped with the other code, and after translation, any single ribonucleotide at a specific location within the mRNA is part of only one triplet. Okay, and the last one is that the genetic code is universal code. Why it is called the universal code? That means the same codons there are 20 amino acids and the same codons remain in every translation process. I think this one. Okay, so yes, this is the universal code, that means the same codon. Yes, I am right. that the same codon is present in every translation process, but there is some exceptional cases that in case UGA and UAG, the two codons, that one coded for pyrrolysine and another coded for selanocysteine. These are the two exceptional codons. Okay, so and another the common codons that are present. So these are all the universal codon because these are present in every translation process. Okay, so here another thing is that why the codon is triplet, why the codon is not duplet. So what is saying that we know that DNA that has ATGC. So ATGC means adenine, guanine, cytosine and thymine. Okay, so if they remain in that like that doublet, okay, doublet way, not in the triplet, doublet way, if they present in that situation, they can generate 4 square equals to 16 different combinations. Okay, 4 square equals to 16 different combinations. How this 4 square is explained here? As 4 kinds of nucleotides taken 2 at a time. So what is saying? 4 kinds of nucleotides taken at 2 at a time. So here the possibility is that 4 square means 16 different combinations. But if the triplet, that means there the 4 kinds of nucleotides but in 3 combinations, or you can say the 3 at a time. 3 at a time. So what is saying that 4 nucleotides and 3 at a time. There is the chance of combination, that is 4q equals to 64. Okay. So in that way this is sufficient. Why? Because to code for 20 different amino acid is possible. Okay. So the combination or they can bind with 20 different amino acid due to presence of triplet codon or triplet code. If they remain at the doublet, so the possibility of the binding of 20 different amino acid is not occur. Clear? So this is the genetic code explanation, why the genetic code is remained in the triplet form, not in the doublet form, and what are the properties of the genetic code I have explained here. So if this is it is just explained very briefly, so I think you can understand it properly. And if you can understand, so obviously comment in the comment section. Okay. And again I am saying that please you just share and subscribe this channel for the growth of this channel. Okay. If you not support me, then how I can help you in that way. Okay. So you just do it if this channel is helpful for you. Okay. Thank you.